So welcome to this webinar on reproducibility in systems biology modeling, BioModels tool. So in this webinar, I will talk about how BioModels, a repository of mathematical models, is playing an important role in promoting reproducibility. So in the talk, first I will talk about BioModels repository itself. Then I will talk about the reproducibility study that we have performed. Right. Mathematical modeling, I think this is where we start our presentation with, because BioModels is a database of mathematical models. So mathematical modeling has become very popular in biology recently. Modelers start to use these mathematical tools to understand the mechanisms controlling the biological system. And this is after physics, which had adapted this tool since ages. And since modelers are more keen on getting quantitative insight, they shifted to apply this on the biological data sets, and try to use these mathematical models, which can answer interesting biological questions. So what is a mathematical model? Mathematical model starts from the knowledge. So we have gathered several knowledge from experimental data sets, and mathematical modeling involves the presentation of this knowledge in form of set of differential equations or set of equations, well-defined equations, which can be simulated to make predictions. And these equations can also be perturbed, can be used to perform in silico experiment to understand the system's behavior better. This has been very valuable understanding several cell signaling mechanisms, cell decisions, cell fate choices, and so on. And these predictions made using mathematical models can be further validated with experiments. This is an iterative process in which scientists take existing knowledge, build a mathematical model, understand the system better, make new predictions, and validate them. And this circle continues until we gain more and more knowledge. So modelers who build models want to share the model with us or the modelers. So to enable that, we have built BioModels, which is a database of mathematical models. As a modeler, you can submit your code of model to BioModels database, which can be then searched and retrieved by other modelers and can be used as a knowledge base to build their own research. BioModels was established 15 years ago to promote easy sharing and accessing mathematical models. This is the home page, which can be accessed using the link shown in the bottom. You can go here and click around and get to know what are the different models available in BioModels. So you can see there are several tabs. Each tab will lead to a collection of mathematical models in BioModels. I will talk about them in details a bit later. So if you're a mathematical modeler, you are building a mathematical model of any formalism. Let it be an ordinary differential equation model, or a logic model, or a Petri net-based model, or a constraint-based model. You can submit all your models, irrespective of the modeling approach, to BioModels database. And you can submit them in any modeling format. You can submit a piece of code in Python or R or MATLAB or Mathematica or any other programming language like C. Also, you can submit your model in standard formats such as SBML, the Systems Biology Markup Language, CellML, FarmML, and combined archives. Although we accept any modeling format, we actively endorse our modelers, our users, to submit models in standard formats so that they can be widely used. So mathematical models are shared and exchanged by modelers through BioModels, but that's not the only service BioModels offers. BioModels also offers data curation. So this is an additional service why modelers prefer to submit their model to BioModels. So what does model curation involve? Let's say there is a modeler who modeled the biochemical network of a signaling cascade, and that's been published. So what BioModels curators will do is that they go to the publication, they look at the mathematical representation of these biochemical network, and encode this in the standard SBML format. There's which is also computer readable format. Once this is encoded, we will run simulation of these models using systems biology software such as Copasi and try to reproduce the simulation shown in the original manuscript. If we manage to reproduce the publication results, then we tag this model as curated, which means that this model has been verified and this model is reproducible and is doing what it's supposed to do. This is a very time-intensive process as we have to read through the publication, make sure that the model is error-free and can be encoded properly and disseminated to the wide users. So we curate models submitted by the modelers to BioModels. Not just that, but we also actively seek out models that is in the public domain and try to curate those models and then put them into BioModels. So while we do that, we try to focus on certain area. For example, in this case, as you can see, we try to enrich model in diabetes. So we had performed this targeted curation project where we actively sought diabetes mathematical models and try to encode them in SBML, reproduce them, and put them in BioModels database, so other modelers can benefit. So if you're a modeler, you're working in diabetes, you can come to BioModels, search the keyword diabetes, and you will find a collection of models that you can take and reuse for your own research. Instead of building everything from the scratch, you have something to work with. We have also done a targeted curation project on neurodegeneration. So we have a very good collection of models on Alzheimer's, Parkinson's, and so on. You can also go and explore this collection. And if you want to know more about this work, you can read the publications related to this targeted curation projects. So currently, we are also doing targeted curation of COVID-19 models. And here we also specifically welcome researchers who have COVID-19 models. If you submit your model to BioModels, we will be happy to cross-verify, make sure that it's working, and encode them in standard format. Also give you the tag that it's a curated model. Right. So as a modeler, you can search and retrieve all these models that we have curated, as well as the models that were submitted by the modelers from BioModels and easily access them. Let's say if I'm interested in immune response, I will go and search immune response in the BioModels search window, and I will get a list of models that are related to immune response. I can click any of these models that I'm interested in to get more and more details about that model. Let's say I click this model of PD1 inhibition of TCR signaling in tumor immune regulation. I will go to this model display page where I will get a basic overview, some sort of description about the model, and the link to the publication where the model has been originally published, and some sort of metadata information that tells me more about this model. It also tells, for example, it tells me that which taxonomy this model belongs to. For example, this model is for Homo sapiens. It also tells me which pathways are modeled in this. You see here that the regulation of T-cell mediated immune response to tumor is modeled. So we add extra notation. We semantically enrich the model with these gene ontologies and other terms so that these models are easily searchable. And this also helps the user to understand the system, the model better. Right. So there are several features that we provide through BioModels for each and every model. For example, if you go to a model, the files tab, you will be able to see all the files associated with the model and the model itself. On number one, you see that this is the XML, which is a SBML file. SBML, for those who don't know, a systems biology markup language is one of the standard formats used to encode mathematical models. It is highly interoperable, which means about 280 different software support SBML. You can, if you have a SBML model, you can import into R, you can import it into MATLAB, you can import it into Python, and there are several simulation software where you can import it into and use this model and try to reproduce the results or build the model any further. So we prefer to curate. That's why we prefer to curate our models in the standard SBML format. So in the file section, we also provide additional files if there are any. In this case, we use because we use Copasi to reproduce the model. We also provide the Copasi file, and we also provide the ZML, which is the simulation experiment description file. to move on further, if you go to the history tab, you can see the history of various versions of the model. So what you see here, these are the versions, public versions 3 and 5. You can go back and download any of these previous versions and use them if required. So whenever there's an update in the model, a new version will be added, but the previous public version will also be retained for general public use. If you move on to the components tab, this gives you a quick overview of the components in the model. You can see that the model has tyrosine protein kinase LCK, and it has LAT T protein and GADS. So you will get all the information related to the proteins involved in the model. That's a quick overview. So last but important tab which you provide here is the curation tab, where you will see the curation the figure that we manually reproduced. So that's a curator who reproduced all the figures from the model that we have curated, and you can see on the right hand side the figure from the publication when you see that the figure exactly matches. And the curation comment tells which figure we try to reproduce from the original publication. So if you come to BioModels and if you find a model curated and you can see the curation figure reproduced, then you can download this model with trust and make sure you can be rest assured that this model is going to reproduce the work shown in the publication. So normally you would expect that, okay, why this is an issue? Now any model should work, but that's not the case. I will talk to you more about this bit later in the next part of the presentation. Okay, so BioModels, to summarize, provides FAIR models, which is these models are findable through BioModels interface, but also through eBay search or MISDI, as well as Google dataset. And these models are accessible. All these models are CC0 licensed, so they are free to use for both commercial as well as publicly. And the code is open source. Not just the models are free, but also the BioModels platform itself. The code base of the platform is also open source, which means anyone can take the code, which is based on JUMP infrastructure, and then reuse it and try to build their own BioModels-like database. As I told you, we focus on SBML, which is highly interoperable community standard. So you can import this SBML model into various formats. And these models are reusable because in the curate models are verified, and you can import them, reproduce the results, and reuse them. So for all these reasons, BioModels is widely used by the modeling community. In a survey where scientists were asked, if you use models in research, which data source do you use? They predominantly chose BioModels in the answer in the survey published in 2017. You can see that BioModels is popular among the modelers or scientists who use models in their research after PubMed and Google Scholar. And modelers also widely use BioModels to share the models. Of course, many of them prefer to share the models via supplementary material, GitHub, and personal website, but a good significant amount of them as disseminate the models through BioModels database. So the content in BioModel had been increasing over time. This includes both submission from the modelers itself and also the submission from the curators in the BioModels team. Therefore, we have a wide collection of models of several organisms, as you can see on the right-hand side, although a predominant of the models are from Homo sapiens humans. And the models and BioModels cover a wide range of biological processes. As you can see here, I'm not going to go into the details, and the models also are covering a wide range of diseases, as you can see on the right-hand side. You can see on the top Alzheimer's, diabetes, and Parkinson's. As I discussed, we have done a targeted curation, try to enrich models in this area, and we are doing several other projects where we'll enrich models in certain specific area of the scientific field. All right, so BioModels content can be broadly classified into two categories. One, literature-based models. Currently, we have about 2,230 literature-based models, among which 960 are curated, and the rest of the models are non-curated. And the other collection is auto-generated models. These are a large collection of models computationally generated through several studies, for example, PAP2 models and patient-specific genome scale metabolic models. So now that we have all these models, some of the users, they didn't want to search models, but they want to search the parts of the model, the components of the model directly. So we built a new service called BioModels Parameters, because we have all these models in standard SVML format, machine readable format, which we can rip these models apart and take all tiny parts, put them together in a database where models can search and find the parts. You can go to the link and access the BioModels parameters, and if you're a modeler, you can appreciate that you can quickly search for a component or a model entity concentration or model parameters or equations used to model certain processes. Excuse me. So there are also quick links. You can go to, from here, you can also go to the original model, and go to the publication, and other resources such as SWRK and Reactor. There are cross references. So here again, you can go and search for your keyword. For example, if you're interested in Cas9, you can search for that, and you will get all the results in which all the models and the parts in which equations in which you have used Cas9. I mean, modelers have used Cas9. This is a very quick way to retrieve the information related to your protein of interest or your compound of interest. Right. So just to summarize about BioModels parameters. So if you want to look at retrieve initial concentration of thrombin, you can get the concentration used by all the modelers. All the used in all the models in BioModels, and you can get a range, and you can use that to study the behavior of your own model. And you can also get parameters. For example, here we've shown the wide range of parameter values used by modelers for KM. In the MAP kinase signaling. Other thing you can also do is that you can take a part of the reaction or part of the model from one model and then integrate it with your bigger model. For example, in this case, we have this TNF NF-kappa B model. You can take part of components from ROS sort signaling model and then integrate it together so that you can now look at the effect of ROS on A20, as shown here. All right. So BioModels is basically helping this notion that's standing on the shoulders of big giant. So if you want to build a model, if you're a modeler, you don't have to build everything from the scratch. You can take what's already there, verified and disseminated via BioModels. So BioModels support model discovery and reuse. Modelers, as a modeler, you can benefit from easy discovery of model. Models are available in standard formats, and the models are curated, which means they're verified and you can trust them and you use them for your own research. And these models are semantically enriched, which means they are well annotated metadata. We add metadata at different levels. At model level, they showed you. We also add metadata at the species level, the component level. So to know more about our work on BioModels and BioModels parameters, you can read these two publications which were published earlier this year. Okay, now moving to the important topic, reproducibility in systems biology modeling. All right. So reproducibility in science became an important topic of discussion recently when published results from many scientific works failed to reproduce. So there was a report, there was a survey a couple of years ago published in Nature, in which it was reported that about 70% of the scientists cannot reproduce others' experiments, and about 50% cannot reproduce their own experiments. Right. So that was the kind of the moment when people realized that there should be an emphasis on reproducibility. And of course, mathematical models, you don't expect them to be affected by this reproducibility crisis because they are mathematical equations, right? So in experiment, you can understand, and also in bioinformatics analysis, you can understand there could be changes in the version of the data. I mean, so reference data and the software and so on. But an experiment, of course, there are many factors. When it comes to mathematical models, then they are mathematical equation, and they should exactly reproduce, and they should do what they're supposed to do. But unfortunately, that's not the case, and there were many models that do not reproduce, and modelers were complaining that they cannot reproduce the results shown in the manuscript. So we wanted to assess what's the status of reproducibility crisis in the systems biology modeling landscape. Right. Before I go and talk to you more about that, I have to clarify these definitions. So there are two things you need to know. Number one is applicability or repeatability. The other thing is reproducibility. So replicability is the ability to use the same code provided in the manuscript using the same software to reproduce the simulation results shown in the paper. So you use everything provided by the manuscript. But that's the replicability. It's also called as repeatability. But reproducibility is different. So it's the ability to recreate a model from the original code without the original code. So basically, you have mathematical equation. You should be able to reproduce it from the scratch and be able to run the model. So we went on and tried to assess the reproducibility. So what we do is we encode model equations. Let's say we take a publication, we find a model, and we encode the model equations in SBML, some biology markup language, get the parameters from the paper or from the author submission. We run the model simulation in Copasi. If the original simulation is done in Copasi, we use different software such as Symbiology, toolbox of MATLAB, or libsbml sim, or Mathematica. So we use different software. So we make sure that the model is actually working. So we will consider a model as reproducible if at least one of the simulation figure from the publication could be reproduced. And of course, we are not very strict. Sometimes we accept minor deviation. Let's say there's some change in the actual graph. We shall accept it if it doesn't affect the main scientific conclusion of that paper or manuscript. When a simulation figure is not reproducible, when a model is not working, we try to fix it. We try to empirically look for errors in the model. Let's say our modeler might have made a mistake in reporting. Instead of 0.1, he could have given reported a parameter as 0.01. So we try to play around and see if there is any problem. We try to fix them. If there's any mistake in the sign of the manuscript equations in the manuscript, we try to fix it and so on. If you manage to fix, then it's good. Then we say this is reproducible with additional effort. And then we also try to contact the authors and see if they provide extra information. When then we try to reproduce the model and then tag it curated, but we don't get response all the time. Okay, so to give you an example, we go to the manuscript. We try to understand the mathematical model and try to reproduce the simulation figures. And then once we manage to do that, we submit this model to BioModels, the curated model, and we also share the curation figure, which matches the original figure in the reference manuscript. So this exercise of testing reproducibility was done along with the regular curation that's being done in BioModels. Right. So in this study, in total, we have analyzed about 455 kinetic models. So this, if you ask us, this is collectively a work of 1,400 scientists. These models were published in research articles from over 152 journals, and they were published from 1980 to 2020. As I mentioned earlier, this study was a part of normal curation activity done in BioModels from 1st of 2018 to 4th of 2020. So the sampling of models is not randomized. So we just follow the natural curation priorities in BioModels. This is determined by the funding. Sometimes we have funding for one project, we curate more models in one area, and also depends on the collaboration and also curators' interest and so on. But nevertheless, the models we cover in this study is about a significant proportion of models in BioModels. As I showed you, there are about 2,200 models in total, and 455 is quite a good amount of that, a good part of that models in BioModels currently. So these models, after curation, after the study, they are going to two branches. If they are reproducible, they go into the manually curated branch. If they're non-reproducible, they go into the non-curated branch. Right, so results. So are the models directly reproducible? The answer is here. You can see about 51% of the models are directly reproducible based on the information provided in the manuscript, but unfortunately, about 49%, that's half of the models, cannot be reproduced using the information provided in the manuscript, unfortunately. So then we try to rescue this model and try to do this extra effort and see if we can reproduce. As I discussed, we will do this empirical correction, try to look for errors in the typos in the model, and we managed to recover about 9% of the model. But then we also tried to contact the authors, and we could reproduce about 3% of the models with the help of others. But the remaining 37% of the models remained non-reproducible. So we have published the study. You can read more. I will also show you about this at the end. Right. So lack of reproducibility is one issue. The other issue we face is the communication with the authors was not that easy. So among the authors we contacted, about 90 authors we contacted, only 30% responded and 70% did not, which is important concern. And even in those cases where they responded, only 13 models could be reproduced and 14 could not. So about half were successful, roughly. So when we looked at the publication and the reproducibility, the age of the publication, the year it was published, and the reproducibility, there isn't a big difference, but you can see that the non-reproducible models are more or less, the proportion is more or less same across several years, roughly. And there was also no clear pattern. We looked at different journals. If one journal is bad and it has more non-reproducible models or not, I think we cannot say for sure, because that's this brought these few journals in the top. You can see they have more of these mathematical models published, and you see there's this roughly equal models which are reproducible, non-reproducible. And there are several journals where we had very few models, so we cannot say that they're definitely good or bad, because the sample size is too low. All right. Moving on. So there are these models which are not reproducible at all, no matter what we do. So what are the reasons for their failure? So when we look at that, we saw that the major reasons were three. So number one, missing parameter values in the mathematical models, and missing initial conditions, the initial concentration of the model entities, and the model structure inconsistency was the third issue. What I mean by this is the model equations are wrong or either incorrect. We know that there are certain terms missing from the equations and so on. So these were the three major reasons we found, and for a major proportion of the models, we cannot even know the reason. Everything is there. We don't know. Things are unclear. So as I told you, where these models are, all the models in the study are in BioModels database. So the list of these models in the study will be out with the publication associated with it when it's published in the journal. So all the models that we managed to reproduce directly or with some extra effort are submitted to BioModels under the manually curated branch, and the models which are non-reproducible at all are submitted under non-curated branch. So these models are not flagged as failed models, but they are simply left as non-curated because we don't want to name and shame at this stage, but we are also aware that there's a need to create community awareness so that other modelers don't pick up the models and spend more time trying to reproduce it. So here the non-curated models doesn't mean that these models are non-reproducible, but it has a mixture of models. Either these models could be non-ODE format, which means we are focusing currently on order differential equation models. There are other model formalism which we haven't yet curated, so they will also be there. Of course, they could be reproducible, but we haven't tested them. And there are many models which we didn't get time to curate, so they're all under the non-curated section. And so non-curated models doesn't mean that they are all failed models. So, but it also contains a good amount of failed models. So we are open to discussion with the community, and then if the community wants these models to be tagged openly as failed models, then we would be happy to do that. So how can we promote reproducibility? There are several publications where they provide several suggestions how to improve the reproducibility. They describe the tools and best practices, and they talk about specialized peer review process for the reproducibility. But what we propose from our experience is that we wanted to keep things simple. We propose 8 points reproducibility scorecard, which can be used by authors, editors, and the reviewers. And these are the 8 points. If you are a model, if you have a model and you need to assess your model against these 8 points and give 1 point when you have yes answer for one of these questions. So the first 3 questions are the following: are the mathematical expression, parameters, and the simulation conditions described in the manuscript or supplementary material. Of course, these 3 were the major reasons why models were non-reproducible. So we asked these 3 questions in the beginning, and yes answer will give 3 points. The fourth question is, are the mathematical model codes for the mathematical expression simulation shared publicly? So it's very important to share the models code publicly. Then, because it promotes reproducibility, someone else can pick up the model and either encode them in their own language or reuse them. So to promote reproducibility, this is very important to share. It's very important to share the model publicly. So we encourage that, and this will give an additional point. So the fifth point is, are the model codes shared in standard formats such as SBML, combined archive? So this is very important because if you have a model which, in SBML format which is highly interoperable, you can run it in several modeling format instead of having to replace or rewrite the code in your own in your own programming language, your own favorite language would be time consuming. So providing in a standard format will make it easy to reuse, and a model which fulfills this criteria will get additional point. So sixth point is, are the model code repositories relevant open model databases? For example, BioModels. So if a model submits a model to BioModels, or there are other repositories such as the Physiome repository or JWS Online, of course, there are also relevant repositories. So this is very useful because these repositories offer curation service, which means there will be someone who will independently verify the model and tag them as curated. So submitting a model, sharing a model publicly is very good. It will give you 1 point, but sharing a model publicly in the modeling database will give you 2 points, because you get 1 point for the question 4 and 1 point for question 6. And then the sound point is about documentation. It's very important to properly document or annotate the model, so that model code, so that others can easily understand and reuse it. And the 8th point is sharing a model results, simulation results. So to verify if the model is reproducible, you need to have some results which is also shared along with the model. So these are the 8 points we propose, and we hope that this can be used by the authors or the editors or the reviewers or the curators to assess the model, so that this sort of gives some sort of credibility on the model for in the context of reproducibility. Right, so we published our work some time ago in late August in bioRxiv, and it gained lots of attention. There were lots of tweets and discussions in the social media and online forums, and it became one of the it became the top 5% article scored by Altmetrics, with a automatic score of 72. So you can see here, this is one of the best BioModels tweet we have done. Right. You can go to this article and read more and learn more about our study, as well as the reproducibility scorecard we propose. Right, with this, I would like to end my presentation and thank everyone. So in EBI, currently we are, it's Henning, who's the head of the cluster, and then Mihai and Tung are the developers of BioModels. Kaustubh and Krishna are the curators. Of course, there were several other curators in BioModels who you can see under the alumni section, who had put several major effort in testing the reproducibility of these models. Of course, finally, the funding agency. Thank you, and happy to take any questions.